Hello, my name is Zoe Lakkis, and I'm from Pittsburgh, Pennsylvania in the United States. 100,000 organ transplants are performed each year, but nearly half of transplant recipients suffer from rejection within 10 years of their procedure. In my project, I used bioinformatics to identify a transcription factor in T cells that can be targeted to prevent transplant rejection. When an organ is transplanted, T cells from the patient's bloodstream infiltrate the organ. Some of these T cells, called TRM, remain within the organ rather than continuing to circulate throughout the patient's body. Because of this, they're able to attack the organ over time and cause rejection. In order to identify a specific molecular mechanism that allows TRM to form, I studied a data set containing the gene expression profiles of different types of T cells. This data set had millions of data points and thousands of dimensions, so my first step was to use dimensionality reduction in order to create a visualization of the data. Next, I used an algorithm called pseudotime, which showed how the T cells differentiated into different states and types over time. This allowed me to identify which cells in my dataset were TRM. I compared the gene expression profiles of these TRM to the gene expression profiles of the other non-TRM types of T cells, and I discovered a transcription factor, which is a protein that regulates the expression of many genes, called PA2G4, that is unique to TRM. Because PA2G4 is uniquely highly upregulated in TRM as opposed to non-TRM types of T cells, it has the potential to serve as a therapeutic target to prevent transplant rejection. My hope is that the discovery of this target will help save many transplant recipients' lives.